In a previous lesson on neurogenesis, we learned that neurogenesis may be interfering with recent memories. In this lesson, we'll look at the flip side of neurogenesis, that while it might interfere with previously established memories, the neurogenesis also might make the hippocampus more capable of synaptic plasticity that underlies new learning. So while neurogenesis can impair previous memories, it can also prepare the hippocampus to make new memories. And again, in another lesson on the engram, the series on the engram, we learned that memories are likely to be distributed over brain regions, but within a brain area, there's going to be a subset of cells that will be allocated to a given memory. And then within each brain cell, a subset of synapses that may be allocated to a given memory. And it may well be that neurogenesis makes new neurons in the hippocampus, for example, and that new neurons are more likely to be allocated in new memories. So physiological measurements indicate that new neurons show high levels of excitability and LTP. Using tagging techniques, new neurons are nearly 3 times more likely to be allocated into new spatial memories compared to older neurons. Thus, new neurons may be preferentially allocated to new spatial memories. The age of new neurons determines their ability to be allocated into a new memory trace. 4 to 8 week old neurons are 2 to 3 times more likely than older neurons to be allocated into a new memory. 1 week old neurons don't allocate at all, presumably because they have not integrated into local networks. And 2 week old neurons allocate somewhat, but less efficiently than several weeks later. At 2 weeks, the neurons may be establishing functional synaptic connections. So by sort of measuring the likelihood of allocation in terms of the age of the neuron, we see a picture. Young neurons don't allocate to new memories. They haven't taken up position. By 2 weeks, they're starting to make synaptic connections, so they're integrating into networks, and then by 4 to 8 weeks, they are more likely to be allocated than older neurons. So let's see if we can visualize what's happening in the hippocampus and the impact of neurogenesis. So the blue neurons here are older, already established dentate gyrus neurons whose connectivity stores older memories. So here we have the blue neurons. They've already they're integrated into a neural circuit. They've already enjoyed synaptic plasticity, and so some of these are going to be involved in storing the memories for recently learned information. Well, along come the new neurons, the green ones, and they're going to take up position in this previously established network. So a wave of new neurons, the green ones, integrate into the network. Now these new neurons interfere with old memories. We saw that in part 1 of this series. But they are more excitable and so have a lower threshold for synaptic plasticity. This makes them more likely to be allocated to new memories. So you see here we have the two sides of the neurogenesis coin here. On the one hand, we're going to be interfering with previously established memories, but on the other hand, these new cells are better equipped to be allocated to new memories. 2 weeks later, a wave of new neurons, now the red ones here, integrate into the network. The green neurons would have been allocated to memories in the previous 2 weeks, thus becoming blue. So let's just imagine the green ones now have become blue, and they're older, and they're taking up positions in the network, and they've already become allocated to previous memories. But we've got the new wave of neurogenesis, and here come the youngsters, the red ones up here, and again, then they might be interfering with previous memories, but they're gonna be better prepared, more excitable, more capable of LTP, so they're better equipped to be allocated to new memories. And the process, of course, is going to be continuing. So neurogenesis is creating waves of new neurons that have this double effect. On the one hand, they're interfering with previous memories, but on the other hand, they are better prepared, more likely to be allocated to new memories. So, the constant wave of new neurons in the hippocampus means that experiences will be sequentially allocated to new populations of neurons, driven by the time scale of neurogenesis. So, as we go through our life, our hippocampus is in a sense constantly renewing itself. And that means that new experiences will be allocating new neurons to be the memories for those new experiences. And again, we should put this whole thing in the context of consolidation. You'll recall one of the reasons to undergo memory consolidation was because of this interfering effect of neurogenesis. But now we see the flip side of neurogenesis. It's also preparing the hippocampus to make new memories. So again, as we walk through life, our hippocampus is changing. It's a dynamic structure. It is making temporary memory traces that can be interfered by new neurons, but these new neurons are better equipped to make new memories. So what is it that determines allocation to an engram? Well, we've seen in previous lessons neuronal excitability, for example, due to elevated Krebs activity. But that neuronal excitability now has two possible causes. It can be recent activity, so cells that were recently active, they're sustaining their excitability, and we saw that that may make them more likely to be allocated to the next memory, so that's how we can link memories. But now we see that recent birth, new neurons are also more excitable. So new neurons are more likely to be allocated to a new memory. Now, the different time scales of synaptic allocation happening over minutes, Krebs excitability happening over hours, neurogenesis over weeks, all of those different factors may interact to allocate complex memories in an organized fashion.